Good morning, everyone. It's a great privilege to give you a warm welcome to NIAB today. The event today is a seminar to celebrate Ian's contribution to science and the impact that this science has made to our sector. It's a sector that in the last 10 years has changed a lot, and Ian's contribution were actually fundamental for that change. I would like to especially welcome Ian's family, John, Alex, and Andrew. Thank you for being here today. And I'm as well today joined by two of my predecessors. I'm Mario Caccamo, I'm the CEO of NIAB, but we have as well Tina and Wayne. And we all were privileged to work with Ian over the many years and see how what he brought to our sector, coming as well from a much broader, if you like, aspect in terms of his contribution to science, was actually fundamental. It helped NIAB to be what we are today, and it helped others. So he worked, of course, with other organizations to be what we are today. As I said, it's exciting times to work in crop genetics and genomics, and clearly Ian's contributions were fundamental for that. We will get a chance today through our presentations to go back into some of these contributions, and I think it's fantastic to see the representation we have today from everyone that has worked with Ian. And I wanted as well to take the opportunity to thank you for coming here, for joining us remotely. And we have really good presence as well from friends and colleagues that are joining us from all over the world. Now, I don't want to say much more because I have here Wayne that will take us in more detail in some of these contributions that Ian made to science. And I would like to welcome Wayne to show me and take it from here. Thank you.
Thank you, Mario. Good morning, and thank you for being here, both physically here, but also online. I think we have a great group of people here to celebrate Mark's contribution. Particularly warm welcome, June, Alex, and Andrew. Thank you very much for being with us today. Looking forward to today to celebrate Mark's contribution. Not just a colleague, good friend, and a good guy. Briefly, Mark's career spans over 40 years in genetics, starting with Birmingham and a PhD in the genetics school in Birmingham, I think with Jeff Gale on natural selection in Papua, followed by nearly a decade as a commercial plant breeder, then director of Lion Seeds for a number of years, followed, I think, with about 5 years, I think, probably in around 2000 with Oxygen, which was a spin out of the Wellcome Trust unit in human genetics. And I think Mark was leading a statistical genetics unit there and made a significant contribution to human genetics, and particularly the early days of quantitative genetics in humans as well. And then I was fortunate enough to attract Ian Mack here in 2005, alongside Andy Greenland and Tina Barsby, maybe a late bloomer. And I think due to their efforts, NIAB is unrecognizable to what it was 20 years ago. Although we see physically some of that difference today, but I think what today will be about is the science and the culture and the research culture underpinning that, with a major focus on translational research, which in many ways was ahead of its time. Before it became popular jargon to talk about translational research, these guys were the forefront of that. So I think it's a great opportunity for us to do this, and thank you so much for joining us today. Can I move the slides forward? Oh, someone I can look at it. Yeah. This is one of my favorite slides of Mark in a flat in Edinburgh, and notice the distinguished bottle of, I think it's port. I think I'm particularly fond of this for two reasons. One is I've got my son Owen on this slide, who is an alumnus of this quantitative methods in plant breeding. I think this tells us a really important story about Mark as well, and that is that the impact of Mark is transgenerational. There must be over 200 alumni, many of whom are in this room today, who were part of that course, which again was set up, I think, Tina, around 2006, and again was recognizing the need for this quantitative capacity in plant breeding, but in biology more generally, and supporting those those individuals wanted to be coached, supported and transitioning into a more digital, quantitative statistical world. So that I think has been an amazing contribution, and I'm sure there'll be people online as well who benefited and have been part of that strong global alumni. I just wanted to throw 2 or 3 slides of some of Mark's impact at NIAB, and of course there are other contributions as well. But this is a snapshot of it, and I think the first thing is that Mark was really at the forefront of really recognizing the importance of historical recombination events, and contextualizing that in terms of plant breeding and the use of historical data. Alongside that, of course, he developed and coined MAGIC, multi-parent advanced generation intercross generation approach, which is shown here, and of course as well has had major contributions to both the analysis in, I think now, more than 50 or 60 crops around the world. So that concept of using multi-parent advanced generation is something which has now become standard, and I think much of that arose from Colin Cavanagh's short sabbatical from Matthew Morrell's lab in Australia, again around 2006, 2007. So a really great contribution to NIAB. This is just a snapshot. We'll hear much more about that this morning. And then he joined SRUC in around 2017. And I want to give a little bit of a snapshot of what he was doing at SRUC in his time with us as well. And I think that John is here. Was John? John recognise this. We have managed to get a group together in Montpellier actually to look at the integration of plant and animal breeding from a genomic prediction perspective, and this is largely Mark and John's work on which appeared in Nature Genetics. CJ will be presenting later as well on designing MAGIC populations, optimizing crossing protocols. And finally, some work as well on GOS. And there'll be talks this morning on some of the work that Mark was leading at SRUC, primarily on how you can use historical data in agriculture that spans, again, plants and livestock. But perhaps one of the things I do want to touch on is this review that was generated with Mark and I think Phil and James here on understanding the basis around 2 phenomena: heterosis and transgressive segregation, which are of course fundamental to plant breeding. That actually, I think, sums up Mark's ability to take many of these concepts and simplify them, but also present an analytical framework for the interpretation and analysis of that work. And in essence, what Mark was describing was, and this goes right back to days in Birmingham in terms of JINX, in terms of looking at the causes of transgressive segregation in terms of dispersion of favorable alleles, but of course as well, what is the cause of heterosis? And one of the frustrations that Mark had as well was a confusion around how we define heterosis, not define heterosis, how we explain heterosis, and the obsession with molecular biologists to bring all sorts of phenomena there, was in many ways, much of the empirical data points to the dominant allele. This work is unfinished, but CJ Young, who talked about this as well, has been following this work, and particularly with respect to a paper that was published around 2017 on heterosis in wheat, highlighting the role of epistasis. And I guess what Mark and CJ were referring to was the phenomenon of phantom heterosis arising from low-density marker. So in this case, there were about 1,600 F1 hybrid parents, 17K in terms of split interrogation, which in terms of the modeling that Ian and CJ were doing, it suggests that this is simply a feature of phantom heterosis occurring at low marker density. The significance of this, of course, is how you start modeling and defining breeding programs to accelerate heterosis in crops such as wheat. So I'm going to stop there. What I want to say is that we miss Mark immensely, but today is about celebrating his contribution. I had a great privilege of working with Mark for over 20 years in different shapes and forms. He was an outstanding colleague, and I look forward to celebrating his contribution with you guys today. Thank you very much.
Thank you. Thank you, Wayne. We will have a few presentations now that will be covering some of these aspects in more detail. And as Wayne said, these are contributions that have made a substantial change in our sector, and it's fantastic. Phil will start with a focus on the magic work, while you're finding the slides.
Yeah.
Yeah. And one point I wanted to mention is a couple of years ago, the NIAB board, recognizing Mac's contribution to NIAB, offer Max a special award that we only give to people that have made that kind of impact to NIAB, and that was being an honorary fellowship. And you will see, if you go back to some of our news, I think it was in 2022, that you will see that special event. And you know, I remember offering this to Mac, and he was really taken and very grateful, if you like, for the recognition. And that day as well, it was a great opportunity for me to understand what dry humor is, and you know what I mean by that. I think I always found Ian's very interesting in his humor, and I learned a lot, not being born here in this country, what it means to have dry humor. Anyway, I'll hand it over to you, Phil. Thank you.
Thank you. Good morning, everyone, and it's great to be here and able to talk to you all. So I gave a lab talk 15 years ago which I called Magic Moments. It was talking people through our first magic populations, and everyone had the classic Perry Como song in their brains for about the next week. So I didn't want to waste that opportunity of giving you all an earworm again. So you'll be whistling Magic Moments for the rest of the day. So outline of the talk. The first few minutes will be unashamedly sentimental, so I do apologise, June, Alex, and Andrew, for that. So my first impressions of this man and fond memories of him, and then delving into some sort of case studies and loose strands of work ideas that we had that we started and haven't been able to finish, but just gives you a flavour of the way his brain worked, I guess. So.
It seems that you're not sharing the Zoom slides. If you can just do that very quickly.
Okay, right.
Just because I think yeah. Oh. It's fine. Yeah. Thank you.
Still checking the band at the top. Okay, so my first impressions were before I'd actually joined NIAB. So he was touting a new project for potential collaborators. I at that time was working for Syngenta as a wheat breeder, and this was an example of what I'm calling Wayne's new NIAB. So raising the research profile. Really moving forwards in research in genetics and breeding, which until then NIAB hadn't really done in any depth before. And he was pitching an idea to use our historical trials data. So NIAB has been running variety trials for as long as variety trials have existed in the UK. So we've got this fantastic wealth of historical trials data. And then the idea was, if you overlay weather records on those, you can pull out what we call genotype environment interactions. So you can find varieties that are more stable across different weather conditions, and ones which are more volatile. And could you then model that for future climate change scenarios? So it was kind of classic Mac. It's looking back at historical data and seeing how we can use that to move forwards. I don't think anything came of the proposal. And I'm not sure that we supported it, but it was an interesting idea. Soon after that, he also contacted us about a new proposal working on multi-parent advanced intercrossing, which is what magic then became magic. So discussing this internally within Syngenta, we thought this was really exciting. I don't actually remember if we formally supported the proposal. We certainly should have done. But we had some interesting discussions about it. So we we looked again at the whole idea of intercrossing different lines. So that way you can stack together different traits and kind of fix those before you go to the field, especially if you've got a decent marker platform. And it's something we'd never thought of before. And then I did apply for a job here at NIAB in 2007, and Mac was part of the interview panel. And my impressions of him were he was really, really bright, friendly, very self-deprecating, and seemed to be obsessed by Lapsang Souchong tea. This was the interview. Wayne was busy in another meeting, but he did come in at the end, and his initial question to me was, "Phil, are you hungry?" Meaning, have you got a passion for this job? And I thought he meant, would you like something to eat? And I very nearly said, no, thank you, I've had some lunch, but I didn't. And then Mac really is.
So if you can click through, you can remove that. Click outside of that.
Outside of it.
Yeah. It's not. Just always fix it.
Yeah.
Yeah, because you can see it. People in remote, you can see it. But...
Well done. Read the title.
You know how to remove it. Yeah, we have some technical.
Thank you very much. Gotcha.
But I'm not taking the. Good.
Right, thank you.
Okay.
So yes, Mac is a colleague, mentor, and friend. So as a colleague, he loved to discuss new ideas, obviously his own ideas, but happy to discuss other people's ideas as well, and kind of build them up as straw men that you could then take apart and see if you could build them back together. So he wanted people to be critical of these ideas, to kind of test them as much as possible before we either dismissed them or carried on working on them. He was happy to accept compromises between perfect, crisp mathematical design and the practical limitations of field and glasshouse experimentation. So he knew that, this is muddy boot stuff. You can't always have a perfect experiment, or if you design a perfect experiment, something is likely to go wrong. So how do you kind of get around that? He was irreverent, often foul-mouthed. He had the most remarkable desk posture. So you'd go into his office and he'd be sat at his desk and he'd be slumped in his chair and he'd be banging away, swearing on his keyboard. It was just, you could hear him from miles away. But patient, really, really good at kind of explaining things, going through data with students, junior staff, senior staff, anyone. Kind, loyal, generous, and modest. And it's a privilege to have been kind of a witness to these formidable partnerships he had with Wayne, first at NIAB and then again at SRUC, and Andy Greenland. This is version, okay. So just a few pictures of that. So this is a very rare picture. We didn't often see Ian in the field. We had a day he called Map the Breeder, where we got our plant breeding colleagues, commercial plant breeding colleagues, and it was the first year we'd grown the magic population as a field trial. We didn't have any funding for that. The NIAB Trust very generously stepped up and funded the yield trials. So it was to kind of generate data for the next wave of grant proposals. And he had this idea that if we go through some of the yield plots as a group of breeders and each make individual notes, do we all pick the same ones or do we pick different ones? And can you actually map breeders as a trait? Which was something he wanted to do as a paper, and it's again a paper that I think never got written, but that's a really interesting idea. This is a fantastic picture. It's Andy and Mac at the end of a long day of seminars. Andy looking as if he wanted the world to open up and swallow him whole, and Mac just kind of being there. And then this was in the pub. This was Mac's leaving do. Really fantastic time. And then Wayne has already alluded to this. So Mac was awarded a NIAB fellowship in recognition of his massive contribution. So he came back in the summer of 2022, I think, for his fellowship, and gave a typically emotional acceptance speech about that. He was such a big softie. E&M Exit Dump is what the beautiful title he gave to the folder he left on our shared drive that had all of his results, all of his analyses, really neatly, methodically organized with a polite covering note explaining where to find each particular thing. So when preparing this talk, I fell down, I opened up lots of folders and just disappeared down all these rabbit holes. There were many failed funding bids. So Mac was a great thinker, but nobody gets every grant funded. And he was upset when a grant didn't get funded, but then he thought of a way to improve it until something did get funded. And the same was true of his papers. So the paper Wayne was talking about on heterosis, I think we had 4 or 5 different attempts at that before it was accepted. I found a version that he called the Quantitative Geneticist's Revenge that he submitted to Nature, and we really got shot down about that. But eventually we got it published. But the main areas of these folders, teaching and training, he did so much, put so much effort into the quantitative methods course. An early advocate for genomic selection, he really saw the potential and how animal breeding lessons can be transferred to plants. Magic and multi-founder populations we'll hear about again and again and again. Genotype environment interactions, well, that was my first exposure to him, and again that kept on cropping up. F1 hybrids and heterosis, I think it's perfect that this meeting is a hybrid meeting. I think that just is excellent. And then this kept on running through. So he'd come up with a scheme, a theoretical scheme, and he wouldn't just like it because it was elegant or because it made good genetics sense, but he wanted to know, okay, how can we apply this to a real world commercial plant breeding situation? So the application was really, really important. This is actually one of his slides I found in this exit dump folder. It's the idea of the multi-parent advanced intercross wasn't Ian's, but he recognized it and beautifully decided, actually, MAIC doesn't really trip off the tongue. Why don't we call it MAGIC? Multi-parent advanced generation intercross. And the name is stuck. Everyone calls these MAGIC populations now. And this is kind of how they work. You take a number of different parents, you cross them in every possible combination to make two ways. So with an 8-way scheme, you have 28 possible 2-way combinations. You then cross each 2-way with each unrelated 2-way, that's 210. And then you cross the 4 ways with the unrelated 4 ways, that's 315. I put my foot down because I was trying to organize all of the crossing. I said, we'll take over the whole greenhouse. We can't grow, we can't fit all the plants in for that. Can we do 208 instead? Oh yeah, of course you can. I've put far too much replication in this. 210 will be absolutely fine. We did a similar scheme for a 16-way. So that's 120 initial combinations. And that was based on a trial design called Latin Square. And we stuck at 120 crosses at each generation. I was doing a crossing program once, and Andy walked into my office. I said, what are you doing? I said, I'm doing one of the crossing programs for Max Magic. And he said, it's just the weird Sudoku. I said, I like Sudoku. Mac likes Sudoku. It's fine, it works. At a key generation, we planted everything out, and these are kind of precious seeds. They're the only seeds in the world of these early generation combinations, and we were growing them out, and a mouse got into the greenhouse and nibbled up all the seedlings. We did have some spare seed, and he helped us rescue as balanced a design as possible. Some combinations we'd lost completely. So we had to try and work out how to rebalance the population from the balanced ones. And because he designed such an elegant scheme, the resulting populations, even given some of these losses, we managed to rebalance them. And it's a beautiful resource. It's balanced. You don't see a skew towards any particular parent or any particular combination of parents. It did what we hoped, which is it gave more recombination than existing populations, which means you get more unique places in the genome to assign markers to, more unique loci. And because it, on the whole, is so balanced, it's allowed many ancient introgressions from wild relatives to be identified. So many of the elite varieties are carrying cryptic introgressions from wild relatives that we kind of didn't know about. And you can pick those up because the map and the population are so good. But of course, there are some loose strands. So the IC in MAGIC stands for intercross. And we were already doing a lot of crossing to generate the populations in the first place. So to make an 8-way, you've got 3 rounds of crossing to make the 8-way. You've got parent by parent to make a 2-way, 2-way by 2-way to make a 4-way, 4-way by 4-way to make an 8-way. That's 3 rounds of potential recombination. For a 16-way, there's an extra layer, 4 rounds of recombination. But that wasn't enough for Mac. He wanted us to keep going. So we booked out the glasshouse for another 4 crossing generations. So we intercrossed. We'd already assembled the 8-ways. We then intercrossed the 8-ways with themselves for 4 more generations based on this 210 lineage model to generate what we called intercross 4 foundation seed. We then need to inbreed and extract a population from that. We did the same with the 16 ways. So this, again, based on 120 lineages. And that's given what we call MAGIC version 2. So 4 additional rounds of intercrossing should be massive, massive recombination between the original parental tract of chromosome in there. But none of this has been funded, so there's still lots to do. We've done inbreeding, but we haven't extracted the inbreds from the MAGIC 2.0, and I'll come back to that a bit later. Once we've extracted the inbreds, we need to line up the original MAGIC with the version 2, the ones that have gone through this extra crossing, and compare the recombination patterns. Has it been as effective as we wanted in chopping up the original parent chromosomes into smaller and smaller pieces? We need to test our hypothesis that this will be excellent for fine mapping and trait dissection. MAGIC is already quite good. When James in the corner over there started QTL mapping in MAGIC, I think typically we found for a good QTL peak, there's probably 50 genes or something underneath it, which is already miles better than existing populations. And it makes finding the gene itself much, much easier, the more defined and more precise these QTL peaks can be. So is that better or not in 2.0? We'd expect it to be. We can look for evidence of directional selection or genetic drift through these extra generations of crossing. And crucially, we can look for novel haplotypes, so novel combinations of genes from the parents, especially around the pericentromeric regions in the middle of the chromosome. These are the parts where you don't see many crossovers at all. So you get these big blocks of DNA that just don't recombine at a high frequency at all. But the more crosses you make, the more likely you are to capture a rare recombination event, which could lead to completely new resources to use in breeding. Another loose strand was this idea of turning a winter population into a spring population. So of the 8 founders we used to make the MAGIC Elite, 7 are true winter varieties. They will not flower unless you put them through a vernalization period, a cold treatment. That makes the switch from vegetative growth to flowering. One of the parents, Cy19, was a facultative wheat, which means it doesn't have a vernalization requirement. Commercially, it was widely grown as a late autumn sown variety, typically after root crop, so after potatoes or sugar beet. It wasn't one to sow in September, it was one to sow in October, November, December. Because one of the 8 parents is different, we would expect a proportion of the population to carry that difference forwards. So to also be insensitive to vernalisation and have a spring habit rather than a winter habit. So while we were inbreeding the 8-way population, we grew some spare seed of one generation in a heated greenhouse. We didn't give it a cold treatment. We had some winters and springs in as controls, and we wanted to get rid of the winters and just concentrate on the springs. So this is the population at the F3 generation in small trays, no vernalisation, and it kind of looks a bit of a mess. But then some of the plants start sticking their head up because they're spring types, they're not winter types. So we go through them periodically every few days and pull out the ones that are extending their stems and move them separately. So the winters stay as kind of tussocky, grassy types. We're not interested in those. We threw those away. Types are elongating, producing seed. They're the ones to carry forward. We repeated that screen at F4 to get rid of any escapes or lines that were segregating and had reverted back to winter. And then we grew them in the field from a spring planting, and we're able to get a small, highly recombined population of around 100 lines that we called SMELs, Spring Magic Elite lines. They're spring types in habit, but they're drawn from the winter wheat gene pool. They're drawn from a different gene pool. We've deposited these as a collection in the Genetic Resources Unit in Norwich, so they're publicly available. I think they contributed to some projects in India, so they were sent to India for testing because they were spring, they're much more portable. But there's still loose ends to do. We can explore linkage drag around this spring habit gene, the vernalisation. We'd expect, because that's the only thing we've been selecting on, we'd expect there to be large chunks of the contributing parent around that locus. In a normal cross, that's what you'd expect. But do you see that in a magic cross where it's had extra recombination? So that's something worth doing. And then this assumption that it's kind of more like winter than spring. So if we have a panel of winter varieties and a panel of spring varieties, and we genotype them and then look at where does it cluster? Does it sit with the winter types but has a spring habit? Does it sit more with the spring types? We'd expect more with winter, but we kind of need to square that one off. Then moving on to the last loose strand, he was dusting off old ideas about how to generate populations as cheaply and quickly as possible. So if you grow a large population, and instead of doing what we call single seed descent, where you have to harvest every plant separately, thresh it separately, sow it separately, it's very labor intensive. What happens if you plant 1,000 lines in a tray and harvest it all together, put it all through the threshers together, get the seed back and repeat? So this is bulk inbreeding. The problem genetically with a bulk is that because you're bulking everything together, you can't trace where everything has come from. You'll have uneven representation from some of the founder parents, some of the founder families. But can we use cheap genotyping markers to extract a population of unique individuals from within that? So we've got this big mass where kind of there's a family structure in there. Can we use markers to extract the unique ones and throw the rest away, the kind of duplicates? And he called this rapid bulk inbreeding. An early name was simple seed descent rather than single seed descent. And he backed it up with simulations. So how big a population do you need to be able to recover 500 unique individuals? Do you need 2,000, 1,000, 5,000, whatever? And how many markers do you need to be able to reliably detect unique individuals? And can you detect outcrossing events that happen during the construction of the population and eliminate those? In the absence of any funding, we've used Rabbit to advance our Magic 2.0 bulks from the intercrossing. We did write this up as a short communication. I don't think it was actually submitted, let alone published. And it was the basis of an extraordinary conference poster. So this was the abstract on the right, and he wrote it in iambic pentameter. I can't find a copy of the poster itself. but essentially this is it, ignoring the book on the left. So it was all about the abstract. He'd illustrated it with some images. There were parts of the sowing, for example, that can be automated. And for harvesting, I think he'd got a picture of some garden shears showing that you can just be as cheap and clumsy as that. And then he'd got some figures for the simulation work that he'd done. And one of the reasons he wanted to use such an unusual submission was that there was a prize at this conference for the most original poster. And the prize was I think it was an iPod Nano, which had just been released. He was determined that this was going to win it, and of course it didn't. But another one of Mac's glorious failures. And I asked him, said, "Well, why iambic pentameter?" And he said, "Oh, have you read this book? It was Vikram Seth's debut novel, and the whole book is written in iambic pentameter." And he lent me his copy, which I got halfway through before I gave up one. So, and that's really all I've got to say. So, a fantastic guy with loads and loads of interesting ideas, far too many for us to see all the way through to completion, but I think we should chase up some of these loose ends at least. Thank you very much.
Thank you very much, Phil. One thing I wanted to reflect is the impact of what the magic population concept has had in science beyond what we do here at NIAB. And I think something which Wayne reflected, which is being ahead of our time. If you go back into where this concept was initially developed, that was before we had any access to proper genomic data, for example. In the last 10 years, we have moved so much in terms of having data that we didn't have before. But being able to see the use of this type of approach to population, thinking what the technology could do, I think was really visionary. Yeah. And it made the concept so widespread today. And as I said, magic is a synonym of what Mac has contributed in many ways as well, since it's associated to NIAB, which we take a great honor to be recognized for. And thanks for that. If there's any comments, or at least, again, thank you for this contribution. Thank you. The next speaker is Rajiv. Is you're connected? Stefan, if you could share the screen. Let me get this. Okay. Thank you. Let me just do this. Okay. If you can put the screen in full screen mode, please. Yeah. I think you have two screens. We see here the presenters. Sorry, yes. If you can change that. Thank you.
Yeah, can you hear me?
We can hear you very well, Rajiv. Yes, we can see the screen, but it's not yet in full screen. And when you put it in full screen, we see the presenters. It's just because you have two monitors, probably, right? It could be like that. If you don't mind, we can go like this. But we see your presenters tools rather than the full screen.
If you click on the display setting, you can change that. It's just right on top. Yeah.
No, I think it's your end, actually, that is the issue. Yeah. Because I'm sure you have two screens at the moment, and you see in your screen the full screen, and we see the presenters tool here.
Okay.
Yeah.
Rajiv needs to go up to display settings.
Okay.
At the top, like his end.
Yeah. If you go to this display settings at the top, Rajiv, you can change that. You have a menu. The second after show taskbar. Yeah, that's what you want. Yeah. Can you see, Rajiv? You have that at the very top of your screen. You have something called display settings. If you click there, you have an option to switch. Yeah, that's what it is, right? Yeah,
exactly.
Rajiv, can you hear me? Yeah. Yeah, that's sorted. Thank you. Go ahead. Thank you very much. Rajiv, you are now set. You can start the presentation. Can you hear me? He's connected. Hello, Rajiv, can you hear me? We cannot hear you if you're speaking. We cannot hear you now.
Can you hear me now?
We can hear you now, yes. Can you start again, please? You can go back into your first slide. Yes, go ahead. Thank you. Hello, we can hear you. You can start.
Yeah, but I can't. My slides are not moving anymore. Yeah, I don't know what happened now. Give me a second. Ah, okay.
Yes, there you go.
Sorry about this hassle about finding out the landscape of how to move the slides now. Yeah. Anyway, thank you very much for giving us the opportunity to present our presentation on breeding for tomorrow, insight from analysis of historical plant variety data sets. So I am Rajiv, and I have another colleague, CJ Young. So we started together in 2019. This is a photograph that we had from 2019 in the dome at Edinburgh, and we, I mean, as Phil was saying about his experience, so we also have gone through the same kind of interview, and Mac was there and was like very I won't say the grilling word, but kind of that one in terms of asking really interesting and to the point questions relative to plant breeding in general, because his passion about plant breeding is immense. So he just a kind of a snapshot of 2019 photograph of ourselves when we were starting there. And this is the list of the Vince and Max group at that time. So Mike, David, Jan Dawson, and all the others that were the part of that group. So our main ambition in principal research group of Wayne in SRUC is plant breeding, sustainability, resilience of the food system across the world. So in the next couple of slides, what I'm going to show you is, as Phil was touching about, Mac's interest in historical data sets. I'm going to give you some examples of that in the next couple of minutes. So I will begin with this interesting slide about the plant breeding timeline. And as you can see that the plant breeding is not only, so when you go through any kind of plant breeding courses, it's always that they introduce you to this, how this is actually been started, how the hybrid breeding started, tissue culture, DNA analysis, all those kind of things. And then recently we moved literally from the whole genome thing to the very precise changes like CRISPR-Cas, and it's kind of immense to see that kind of growth. But as a variety of data sets that we are going to use to display to you is, how the plant breeding works. So most of the breeders, they got the breeding lines. They have to go through a lot of year trials, and then only the handful of them make through the national list, and only few of them make through the recommended list. So now we ask the question that, given that there is a huge competition of the breeding lines, and only the handful can make it through, there must be a merit how they have gone through that kind of a cycle. And you can see in some cases, some varieties, they are there for a little bit longer than the others. In other cases, some varieties, they cannot be on the recommended list for that long. So can we use the age of the variety to look at the historical data set, and can it pick up some signals at the genomic level using GWAS? So that's the notion that we started with. And given that variety trial is kind of a survival of the fittest. Only the most resilient and exceptional plant varieties withstand the rigorous selection process of variety registration. And then it's quite a cutting edge competition. It's not that only the ones that withstand very well in that competitive adaptation can make it through. So in the next couple of minutes, I'm going to give you some examples from wheat, barley, and also another data set, which is probably not for the plants, but from the cows. And I will give you the information that how this can be used across the systems. So for the wheat, we have used actually the winter wheat, and also we use another complementary data set that comes from Brazilian spring wheat. Another one that we have from barley is basically the two variety data sets from the winter barley, spring barley, and the cows data set that I'm going to present is the Lang Hill data set that is quite unique in itself. I will give you a little snapshot of that in the next couple of minutes. So when you look at, as I was saying in my previous slides, look at the variety yield data sets of the varieties that got released in the last several decades, you can see that there is a direct correlation between how well they perform and the year of release. So, for example, on the top is UK winter wheat, correlated quite well with the yield, with R equal to 0.94. Similarly, you can see the UK spring barley, which also is quite well correlated with the year of release. So that actually led us to the question, can we use straight away instead of, if we don't have a historical data set of yield for all the varieties, can we use the years as a proxy to pinpoint those genomic regions that were selected over the several years? So that's the interesting question. So here is the GWAS peak of the UK winter wheat varieties, and on the lower side is the Brazilian spring wheat varieties. Both data sets have got quite a reasonable amount of variety in terms of the numbers, 400 plus in case of UK winter wheat, 300 plus in case of spring wheat varieties. But what you can see here is it actually picks up quite interestingly a lot of loci on the whole chromosomes. So in case of UK winter wheat varieties, there are only 9 regions. On the other hand, in the Brazilian spring wheat varieties, there are only 2 regions. And then we ask the question, is there a congruence in both the regions? But unfortunately, we couldn't see anything because they are 2 different data sets, and probably they have been selected differently because it's a spring wheat in case of Brazil, and it's a UK winter wheat. So there are 2 different data sets, probably though, and they are adapted to probably different regions. So, but it's interesting to see how many of these are actually being picked up because they are linked with the yield or with the disease resistances. So I'm going to give you another example. Here is the example of barley. So in case of barley, what you can see here is, sorry, in case of barley, what you can see here is that, give me a second. So in case of barley, what you can see here is that we have GWAS analysis on the spring barley and the winter barley separating out in UK, and both of them pick up some of the different regions, but one of the and many of them are actually the flowering time genes. Interestingly, one region that we pick up on chromosome 5H short arm, which I have marked here with question mark, is quite interesting because this region is actually present across winter and spring gene pool or spring and winter varieties, and it is to do with disease resistance, powdery mildew disease resistance, which is quite remarkable to observe just utilizing age of the variety as a trait. Then we also asked the similar question in Lang Hill. So Lang Hill is a cow breed that's been bred as a study at SRUC, where they have two groups. One is called control group, another one is called selected group. Control group is actually been inseminated by the national average group in case of control. In case of selected, it's the top sires of the national of that year. So, and then what you can see here is the milk and the fat protein is between the two groups is also separating out quite well. Then we asked the question, can we use this year of the birth of the cows as a trait to mark similarly as a variety in plants regions? And what you can see here is this is the snapshot of the 29 chromosomes of cows, and there is a nice peak on chromosome 3 that we have picked up. And similarly, we try to use different ways to see whether this is because of the control and selected group analysis together, or we could see any differences if we actually use that as a cofactor. We couldn't really see much changes because what you can see here is still the chromosome 3 has been picked up quite nicely. And so, and then this region is a milk protein percentage in German Holstein cattle in the literature, which is quite interesting. Then we also try to look how these alleles are separating out when you look at the years. So here, what you can see here is that old alleles are quite well separating out from the modern allele. So modern allele is somehow been picked just recently. So it's not like, you know, you have only one allele being selected on those loci for very long, and then suddenly the modern allele started selected by the breeders. So that's quite interesting to observe. Then we also try to look at Eigenjivas, another way of finding the peaks without utilizing the traits. So in here, we use the 10 principal components in wheat. A, B are the wheat example from Brazilian wheat and the spring wheat. Similarly, we also use the C and D. These are the examples from the barley, winter barley and the spring barley. And what we could see here is that in case of wheat, we do see that these PCAs are actually been, jivas of the PCAs are actually been picking up the historical introgressions. On the other hand, in case of barley, because barley has never withstand any kind of introgressions in general compared to wheat, we couldn't really see anything common across the gene pools and across the principal components either. So which is quite interesting to see. So finally, I would like to conclude that the jivas of age that's we have shown with wheat and barley that it could actually pick up nice yield-related traits, also the disease resistance in case of barley. And Eigenjivas is another way of looking the historical data set when you have the marker data set but don't have the traits. But in case of wheat and barley, again, it is not picking up the similar things because barley has got no introgressions. On the other hand, wheat has got introgressions. So you could get 1B, 1R, and all other kind of interesting introgressions that you pick using Eigenjivas. And finally, as you can see that we have extended our analysis of age into the livestock. So this system can be used anywhere where even the data set is not available. All these are actually been published in TAG already, and the multi-parent population has been used as was told by others that in wheat to validate many of those peaks that I have shown in my presentation. And thank you for listening. I'm now handing over to CJ. Yeah.
Thank you. Thank you very much, Rajiv. And I'm aware as well that we have many people joining us remotely. If you have to, if you want to comment or ask any question, please unmute yourself. But thanks for this presentation. I think it was a great example in where we can see the interface looking into future approaches in where the contributions we had from Ian's work set the foundations. And in particular, I think it was interesting to see how we can move from some of the systems we are using in crops into animal genetics as well, and some of them move through this boundary very well. Some of them needs adapting, and it's something which Wayne mentioned as one of the key contributions that Mac made in his career. Now I would like to hand it over to Wayne, who is going to chair the rest of the session in the morning.
Is you coming in? Briefly?
Yeah,
sorry.
do I still have time to continue?
I'm sorry about that. Sorry, I thought that was the end of the presentation. Sorry, my mistake. Go ahead, Sisha. Sorry about that.
Okay, cool. Yeah, I'll try to be quick, realizing the time limitation here. So basically here, I'm going to talk about a different project on looking at the UK plant variety recommendation system. For those of you who know Ian, you will have known that this topic is definitely in one of his high priority lists in terms of his area of research. So as Rajiv highlighted earlier, the process of getting varieties or lines from breeders to farmers is quite a long process. You have to go through things like national listings and recommendation listing in the UK, and you end up with only a handful of variety being recommended every year. And this generally applies to many of the major crops in the UK, such as wheat, barley, and oats. And it tends to focus on all the key traits such as yield, disease resistance, as well as other agronomic traits. So in this slide, just kind of show you how the recommended list looks like every year. You get a PDF or Excel spreadsheet that gives you the variety performance against the control variety. I think in the last couple of years, they developed this online variety selection tool where you're allowed to pick up the data according to what you would like to compare with. For example, you could pick data from 1 or 5 years. You could pick data from your local trial site, regional means, or even overall nationwide means. And basically, these are just helps to help guide growers and farmers to decide on which variety they're going to plant for the next year. As I mentioned earlier, there's this concept of getting regional means, and this figure on the left here is just showing you how the regional means is being calculated. You have the whole UK is split into 3 regions. You have the north, you have the east, and you have the west regions. As you can see in the figure in the middle here, it's showing you the climate variation throughout the whole countries. As you would expect, going from north to south, you get increasingly warmer temperatures. You have a smaller temperature range in the coast, as well as less likelihood for frost. And in the northwest part of the country, you tend to have a lot more rainfall as well. So in some sense, the point of this climate figure is just to show that the regional EU classification has kind of no environmental basis to it in some way. Then going forward, there's really a lot of challenges in trying to predict variety performance over the years. As you can see in the figure on the left here, it's just picking out 2 different years, trying to predict the variety performance. First in 2007, as you can see, the predicted versus the observed performance were very well correlated. So if you're using, if you're a farmer in 2006 trying to decide on a variety in 2007, you'll be doing really well. However, in 2016, as you can see, the performance is really bad. No matter which method that you choose, the prediction accuracy is really low. So in 2016, you end up most likely not be planting the best performing variety that you should be. And so according to the variety selection tool, there are kind of like 6 different ways you can choose a variety. You can choose it based on individual trial sites. You can base it on regional means. You can choose it based on all trial means. And it can be taken from 1 or 5 years of data. And for some of you who have heard of Ian's talking about this before, it basically boils down to the concept of the use of blue versus blob, and how we can switch from the current system, which is based on the blue or the simple mean system, to a blob by simply treating the variety as a random effect. And on top of that, we're also interested to test and see if the use of genomic marker can improve the prediction as well. Just here, it's just kind of a slide to show the prediction performance in a couple of trial locations across all the different methods that were tested. I'm not going to go into the detail here, but just to highlight that in some year, you can see a clear improvement by using the blob approach as well. Again, this slide is a bit hard to see. So the next slide is just kind of a summary of all the correlations across each of these methods. And I just want to point to you the one at the bottom, which is A5B, which is the approach using all trial sites across 5 years of data with a blob approach, produce the best correlation of all methods. So it clearly highlights the improvement you can get by using the blob approach compared to the currently available simple means. And on top of that, which is kind of surprising, is that a lot of the trial performance in the UK are in fact better predicted by 1 single year of data. If you can look at just within individual sites, the 1 year of data predicts better than the 5 years of data, and so on. And in next, I think this is one of the last slide. We're trying to quantify what it really means in the yield deficit. So how much yield are we losing by predicting the wrong variety? So the way we quantify the deficit is to take the difference in the yield of the observed best variety against the yield of the predicted best variety and divide it by the maximum possible range from the observed best and worst variety. As you can see in this figure, there's not too much difference of whether we use the existing method or versus we use the blob approach. The improvement is very slight, very little. And in fact, in some counties that are highlighted in this map, they benefited from using the blob approach and some didn't. So overall, we find a very minor improvement, small but significant improvement. And really going forward, there are really many other ways we can improve the approach that we're using to recommend a variety here in the UK. First of all, there's a lot of trial data coming from the national list as well as unreleased varieties. To be able to include that would definitely improve the prediction performance. And on top of that, to be able to also include data from breeders who have run a lot of extensive trials for their candidate varieties, that would be really helpful. I understand that this can be challenging due to data proprietary issue. However, I think later on today, you're going to hear more about data encryptions and the use of it in plant breeding from Richard. And lastly, it would really be great if you actually have replicated data from each of these trials, which at the moment is unreplicated data. And just want to highlight that a previous paper that we published on the plant variety registration system, where we highlighted the importance of the use of genomic marker in DUS. And to be able to have genomic marker for not just for DUS, and it can also benefit the use here, as we showed by using the blob approach with the genomic markers. So essentially, it's kind of a multi-purpose use of data. And on top of that, if we have replicated data, we'll be able to try other blob models with the use of things like factor analytics, random regression, things that are essentially to help us improve our modeling of genetic bioenvironment biomanagement interactions. And lastly, there's been a lot of development in machine learning-based approaches. It will definitely open up a lot of opportunities to improve the variety recommendation systems. So with that, I'd like to thank everyone for listening, and really a big thanks to Ian for being a great advisor in the past 4 years. So, thank you.
Thank you, CJ. If there's no any comments, as I said, anyone connecting as well remotely, if you want to ask a question or comment, please unmute yourself and go ahead. Thanks. Thank you for this. I'd like to hand over to Wayne to share the rest of the session. Thank you.
Thanks, CJ. Thanks, Rajiv. We'll move on to the next presentation in a moment. I just wanted to offer a couple of reflections on what we've just heard and Ian's time at SRUC. I really know that we push for time now, Mario, but I just wanted to say a couple of things I should have said in introduction, actually. One is how pleased we were and how thrilled Ian was to be awarded a chair at SRUC, and I think that was something that was really important. And that, of course, gave Ian the opportunity of influencing activities and research, not just across SRUC, but much more broadly as well. The other thing that I think we tend to forget, but during Ian's time with us at SRUC, it coincided with COVID. that period of COVID was really a period where we all had to adapt and change. And one of the things that we decided under Mark's leadership was that we'd focus on the use of historical data. And that's what I think we've seen here, is that how that is reflected, but also using that historical data to be looking forward into a world of regenerative agriculture and how you can design idiotypes for sustainability and circularity in agriculture. But I think the other thing that we wanted to add to that was really a flavour of Ian's creativity, imagination, and indeed resilience. Right up until a couple of weeks before he passed away, he was online, he was leading this research group remotely, and I think his diligence and dedication was quite incredible. So I'll now move on to the next session, and great pleasure again, and it's kind of quite a nice segue into John Hickey. Many of you will know John. John is currently head of corn product design at Bayer CropScience. John, you may well offer a few comments, but I think I know you're a good friend of Ian's, and maybe you first met in Australia. And I know then continued your collaboration at Roslin, both here when Ian was at NIAB, but also in Roslin as well, a number of really influential papers in terms of simulation and breeding design. And thank you very much for being here today.
Thanks, Wayne, and thanks, Andrew, Alex, and June for being here. Like everybody in this room, Ian was a very good friend of mine. He was a very good friend to me. I first met Ian in Australia in 2011 when he came to teach his course, and it was my first exposure to plant breeding. I was an animal breeder. If I hadn't done Ian's course, Ian hadn't lit that spark for me, I would have had a very different career. I moved to the UK in 2013. Just before I moved to the UK, I didn't know anybody in the UK. I sent an email to David Marshall, Ian, and Fred Van Eyck in Wageningen saying, I'm John Hickey. I met some of you in various places. I'm moving to the UK. I would like if you could help me integrate a little bit into the plant breeding community in the UK and Europe. And all three helped, but really Ian helped in a way that was unbelievable. Within 3 weeks of arriving in Edinburgh, I had got a slot to speak at Monogram. The agenda had been closed probably 6 months, a year before, I don't know. But somehow Ian managed to pull some strings, and I got a talk in Monogram. He brought me in, he brought me down here many times, brought me to all the breeding companies. We went together to all the different breeding companies. He introduced me to many people in this room, many other people, Wayne. And he did all this. We had an awful lot of fun doing all of this, because many of you know Ian for a lot longer than I know him, and knew him maybe in a more personal level. But he was always laughing. Everything was always funny with Ian. Himself would be the center of his own jokes, or whoever was not there, or whoever was there. It would all be and always done with deep intellect and good intentions and good nature. And this is a photograph from Portugal, I think from about 2017. And this morning I had a flashback, because this was how I used to meet Ian. I would get up at 4 o'clock in the morning, get some Ryanair plane, usually from Edinburgh, usually to Stansted. Ian would collect me in his big car. And Ian, as we all know, was a very humble individual. And he used to have this big car. So I used to always tease him, say, Ian, like, why do you have such a big car? And he always claimed that it was Alex's fault, that Ian said, I don't really want this car, but Alex, he keeps buying cars and he keeps making me drive them. So, and another thing about this photograph that, at least for me, is always how I remember Ian, is not the way he is in this photograph, because in this photograph, both parts of that shirt are sticking out. And normally, Ian, that's a typical, for me, the typical Ian stance, but usually one of those two parts of the shirt will be stuffed inside the trousers, and the other one will be outside the trousers. And Ian, for me, Ian was a real, and again, many of you know Ian in a different, maybe deeper way than I do, but from a distance, from where I stood with Ian, this was a person with a real intellectual, right? He had deep thoughts about many topics. He seemed to have an encyclopedic knowledge of the scientific literature from 100 years ago up to today. And he could connect everything, new ideas, old ideas, ideas from animal breeding, plant breeding, quantitative genetics, statistics, evolution. He was able to bring it all together. And Ian encouraged everybody around him. I was one of the young people that was in his sphere of influence. Many other people here today are, other people were too. And Ian really helped us all to kind of have the confidence to do what we could do. And that for me, it will be his biggest legacy, because I think many of us have careers and had impact and had opportunities because Ian helped us have the confidence to believe in ourselves. And I think he's a role model for all of us. So I do have some slides about some stuff that I can talk about, but that's mostly the most important thing I wanted to say, right? So I work at Bayer. Bayer is, well, it has an important role in the world, like all of plant science, because natural resources are under pressure, and we need technology that can increase the efficiency with which we use those resources and produce more output from those resources and protect and as Wayne said, regenerate those resources. And when Ian probably started his career as an animal or as a plant breeder, in some sense, a plant breeder did everything. They were a one-person, multifunctional machine. And I think the journey of the technology in the last years, and perhaps not just in Bayer, but definitely in Bayer, has been a journey of specialization, where in the old days, Adam Smith, an individual person, I think they could make, I forget these numbers, but certain small number of 200 pins a day one individual could make, but a team of individuals, I think it was 20 pins a day or something. Yeah, something very small. Anyway, when you put a team of individuals together who have specialization, you can have a massive increase in the productivity of that group of people. And also you could have a massive increase in the innovation that happens within each of the elements of constructing that pin. And I think that's been Bayer's journey in the last 30 years, about having deep specialization in each segment of the plant breeding process, and then having the expertise and the time and the focus to really innovate in the particular element that you're responsible for. So Bayer is a very large company, 100,000 people, 3 divisions: CropScience, pharmaceuticals, and consumer health. We spend 5.4 billion on R&D, and we have 44 billion in revenue. If we look at CropScience itself, it's about a third of the company, half the revenue, slightly 40% of the R&D budget, and a third of the employees. The cell of Bayer is to be the #1 technology provider to the global crop sector, with 3 big platforms. So we have what we call seeds and traits. So seeds are the germplasm that we seek to improve and deliver. Traits are the genetically modifications that we add to that germplasm to give them particular features, like resistance to particular disease or resistance to glyphosate, as an example. We have crop protection products. I think we have the biggest portfolio in the industry, but we have also lots of little other elements that get added to that, such as seed treatments, microbes, etc. And we have a digital farming platform, which is essentially ties together our data, together with any data that's available in the public domain, together with data that each farmer generates through their day-to-day processes as they operate their farm. And then all of that can be brought together to provide recommendations, tailored solutions, essentially the recipe for the inputs and outputs of each square meter of soil in your farm. If we look at the breeding piece of Bayer, so this is just the row crops. There is a whole separate division that looks after vegetables, which has now a close relationship with NIAB, as we know. But in the row crops space, Bayer products impact 180 million acres of crops globally. We have 6 major crops: corn, soy, cotton, canola, OSR, wheat, and rice. And yeah, globally, and probably the largest provider of seeds. The journey of Bayer's activities in breeding has been really going on since Monsanto entered the seed space in the mid-1990s. Essentially, we have 4 evolutions. So breeding 1.0, breeding 2.0, breeding 3.0, and we're now in what we call the precision breeding era. Breeding 1.0 was really about spending more money than anybody else on breeding and doing all that you could with the technology that was available at that time to be faster than anybody else could be. Breeding 2.0 was about increasing the accuracy of our data generation, so getting really good at field testing at scale. Breeding 3.0 was bringing in better analytics into the decision-making process, maybe more digitization of the whole automated machine that delivers the breeding program. And precision breeding, which we're now in, is about it's being customer designed, and I'll say a bit more about what that means in a minute. So this is breeding 1.0, the classic breeding program that everybody knows. Doesn't work. The one at the top there, the green, makes some crosses, put them through a whole series of tests, and comes out some varieties at the end. Breeding 3.0, it inserted this red and blue step in between, which essentially involves genotyping individuals and advancing individuals based only on their genotype, but still following those individuals all the way through the testing process. What that allowed the company to do was to essentially have an 8 to 10-fold increase in scale, take a year or 2 off the breeding cycle, and achieve a 30% increase in the rate of genetic gain. This is a nice picture that sort of captures that. So we eliminated what we might call the first stage of testing, screening 1, and we took that into the lab in our Chesterfield facility, where we could essentially represent all these acres or hectares of field testing in a tiny microchip. Precision breeding is about, instead of from finding the best to designing the best, and this is a sort of a comment that would be fun to discuss with Ian, because he was very philosophical about plant breeding. In some sense, old plant breeding was anomaly detection. You test as much as you can and detect the anomalies that are there, and use statistics and methodology and the kinds of things that Ian developed in the magic, as an example, to kind of generate more of these anomalies and efficiently detect them. What Bayer would like to do with precision breeding is to move that from an anomaly detection to actually a design that you're driving at. The first way we get at that design of that output is to really precisely try to capture what it is that the customer needs, what it is that the customer might want, what would drive profit on a customer farm, really understand the economic drivers of profit for each farm and for each system and for each square meter of soil. And we built a system in the last few years that really, in my view, is a step change in how the plant breeding industry tries to get to that level of understanding the customer. The next element of precision breeding is something that, of course, is well known to many people. It really is about leveraging our ability to measure the genome to its fullest potential in a breeding program. So Ian worked in oxygen. He also, made a lot of contributions in plant breeding in the last 20 years, where he was leveraging the ability to measure the genome precisely. And this is a picture of a human whose genome was measured by 23andMe, and we're able to say things about this human. So we can, in this case, color their ancestry by which parts of their genome came from which ancestry. But you could, and 23andMe do this, you can layer on top of that predictions about, based on the measurements of this genome, we can say things about the phenotype we would expect that individual to express, its height, its weight, its risk for a certain disease, etc. And genomic selection is the technology that allows us take that forward. And genomic selection is a very simple idea. It's, measure the genomes and phenomes of this set of individuals that are a training set, calculate a prediction equation, then combine that prediction equation with the genomes that have been measured on unphenotyped selection candidates, use that to identify who are the best ones. So you can select individuals directly based on the measure of the genome rather than on their phenotype. And I think this might be, it's this slide. So about a year and a half ago, I gave a talk at a conference in Paris, and with Bayer, it's a bit of a process to get your slides approved. So generally you just get some dump of slides that have been approved and you just present them. And in my slides, I added one slide. This was the one slide I added. And I got down from the podium and I sat down and I opened up my phone and I had a Twitter message from Ian saying, "John, you can't say that." So, what this allows us to do is select directly on the composition of the genome, and it's the biggest technology leap in the 10,000-year history of agriculture. Ian didn't agree with that. He said it's the second biggest. In his view, the biggest was hybridization itself, that was actually a much more fundamental side. And Ian is right, but it doesn't suit my story, and I already got the slide approved, and I don't think investors want to know about hybridization from 10,000 years ago. How am I doing on time, Wayne? What?
You need to check your phone.
How am I doing on time, Wayne?
I think another couple of minutes, I think.
Couple of minutes, okay. So what we've done with this precision breeding then is essentially reorganize the way plant breeding programs are structured. So in the old days, you had this process that would make crosses, put them through a series of tests. It produced commercial varieties. Then these late stage individuals from late in this testing process would get recycled as the parents of the new population. That allowed us to do one cycle of genetic gain per unit of time. So for example, it would take 5 years to do one cycle of genetic gain. This decoupling process allowed, what we're doing is we're taking this recycling piece out and we're putting it into a process called germplasm improvement, which we run entirely on genomic selection. We still maintain the product development, product evaluation piece as a separated coupled process or decoupled process. And that simple concept allows us to achieve 15 times more cycles per unit time. And given the breeder's equation, which Ian was very fond of, time is a fundamental element of that. It's probably the most untapped element in the breeder's equation. This is a step change. In the bear breeding programs, we think it can deliver 2 to 4 times more genetic gain per unit time and unit cost. And this is a very simple concept, and who knows where its original source was, that concept. Certainly we all did a paper together that I think laid it out in 2017 through a project that was done jointly with Ian, Roslyn, Phil, many other people from NIAB, and the wheat breeding companies around about Cambridge that Ian, brought together as a consortium, and it's easy to make big claims, but I think as a paper and as a group of individuals working together, this may have been a very important project for the history of plastic, and it wouldn't have happened without Ian, because Ian was the glue and the catalyst and the encourager that brought all of us together to do that. So when I'll probably stop here, I've got more canned slides from there that I could go through, but it's just more of the same, right? So, thank you.
So I think we have a couple of minutes of questions before our next speaker. So maybe we'll kick off in the room here and online. I'm not sure we're getting access to online questions, Mario. Let's start in the room. Any questions?
Right, John, this point you made about finding the anomaly, which has been the historical way of approaching breeding, moving towards what I will interpret introducing that anomaly, because that's what you're saying. We will be targeting our approach. There is a fundamental difference. When you find the anomaly is in a stable system. When you're introducing that anomaly, you not necessarily are doing this in a controlled manner. I wonder how much this you see as a challenge. In my mind, the technology here that could drive this is clearly gene editing. I think that's probably what you have in mind. But
yes,
I say, kind of looking into the future, how much you think that is the future, and how much of that introducing something that is not in a stable context is going to be feasible? It will deliver its promises.
So I think there's two elements, Mario, to where you're going. I think with your comments. One is, in some sense, with this genomic selection, we are assembling together the anomalies. And I think the concept that Bayer has, that it's subtly different, I think it's just an importantly different mindset, is that rather than just chucking the anomalies back into the sausage machine and let them spit out more anomalies, it's really trying to bring those anomalies together in a designed way that's got the customer, the quantification of the customer's profit in mind. So that's one element of, and that's recurrent selection. And then to your point on the gene editing, I completely agree. I think gene editing is fundamental. And Ian was great to talk about all these topics with, but breeding has been, the rate limiting factors. We have no control over random processes, recombination and mutation. With gene editing, we can get control over where we want the mutations to be very precisely, and we can probably get control over where we want the recombinations to be. And I think this precision is going to be a huge opportunity. As we all know, it's going to take a lot of investment in biology to give us the knowledge we need to no longer just lean on the crutch of randomness.
Any questions online, though? You would see. Yep. Richard?
Hi, James.
Yeah, you were talking about Reed's equation, and you said that time might be the part of that equation that's the most prominent for improvements at the moment. So I remember Ian was always interested in something called captured called so basically going from gamete to gamete in a petri dish, basically. But if you get the untestable ability to go from gamete to gamete in petri dish, so you can incorporate costing. So just really rapid, breeding and shortening over time, and then incorporating development selections within that technical development. Is that anything that's on your rate or Bayer's radar, or is it feasible? I mean, I know there's probably technical issues with introducing wanted variation because of the techniques you may need to.
Because of the
techniques you might need to employ, like just steps like that. But is that anything that's ever been raised or signed in
So conceptually, yes. In practice, no, for reasons that are shared. So we can do 3 cycles a year in a glasshouse with pretty conventional technology. Right? And when we spin genomic selection at that speed, the accuracy decays. Every cycle it has, right? So if you spin too fast, your accuracy decays before you can retrain your model, because you can only train your model once a year, but you can spin 3 times. So we've chosen at this point in time to make it work with conventional technology of spinning 3 times a year, because we're worried about this decay of accuracy problem. But I think if we demonstrate that this can work, I think, yes, the obvious next thing to do is to go after 6, 8, 10 cycles a year, because it unlocks an awful lot of things. But yeah, we're quite a while away from being able to take on that, I think.
John, maybe I'll ask a question, and really you wear, I guess, both a science hat in Bayer and you have to wear a business hat, right? So what is Bayer's business? Can you share, or can you reveal how Bayer sees regenerative agriculture, and whether it sees that as kind of a business opportunity?
Yes, for people that are interested, so the Bayer is 30,000 people, so we all have our domains in which we're responsible for, right? And I'm responsible for quantitative genetics of making this thing work. There are other people that are much more could answer that question much more deeply, but I have seen them talk. Actually, I've probably seen the same information that anybody here could see. Bayer did a very interesting innovation summit in New York autumn time last year, and I'd recommend anybody that's interested in thinking about regenerative agriculture or sustainability. It's really good. In my opinion, it's really good stuff. And the core of it, they have some key sentences which I can't recall, but the core of it is do more with less while regenerating more. So, yeah, what can I say? When the Bayer technology, not just Bayer, but many companies, the technology, it does a lot to regenerate. Like when you spray glyphosate, you don't need to plow. That has a huge amount of positive impacts on the regeneration. And in that innovation summit in New York, they announced that they saw an opportunity to double the sales potential of Bayer CropScience. And I think if I'm not mistaken, the number was 100, it could be 100 billion, and it could be 200 billion, or it was some huge number like that, but doubled through this opportunity that regenerative agriculture offers. So it's a serious business opportunity.
Thanks, John, and maybe that's a lot to do with seed as well, so we can maybe pick that up over lunch. But thank you very much.
Can I ask a non-scientific question? In that picture, so you have Ian and you, the other person there is Pancho.
Yes.
Pancho Crosa.
Yes.
I can only imagine. Can you reveal what you discussed that day? Because I guess, if you can maybe say briefly who is Pancho, because they work together very well. And,
sure. So Pancho Jose Crosa was the biometrician, head of biometrics unit at CIMMYT for 40 years. I think he moved there in 1985. He retired in 2012, 13. I approximate that he is about 77 years of age, and he still comes to the office every day. He's deeply interested in science and football, and maybe a bit like Ian, intellectual guy that's maybe not very reverent, again has had a huge impact on low and middle income countries, and Ian was working together in the last 10 years, right? Well, not 10, 5 years or so, right? So,
okay, join me in thanking John.
Thank you.
Mario's comment is a kind of a nice introduction to our next speaker, Sarah Hearne, who's joining us from Mexico. I think it's probably early in the morning in Mexico. Sarah, so thank you very much for joining us. I'll pass over to you now, but thank you so much for presenting.
Hey there, everybody. Nice to be with you all. Can you, as usual, as we ask always when I'm joining in from Zoom, can you see my screen?
Yes, we can. Thank you, sir.
Yay! Well, excellent. Okay, and you can see it in the right view, which is always a challenge. So yeah, I'm joining you from Mexico. It's 6 a.m., just after 6 a.m. here, so you get me bright and shiny. I haven't been with you up until quite recently. So yeah, apologies if some of this is repeated. So I had a history with Mac from time in the UK, coming in and out of NIAB for visits when I was with the CG system, both with CIMMYT, and previous to that with IITA, where I was based in Kenya and Nigeria. So lots of conversations with Mac, pre-magic population conception, post-magic conception. And then in the later years, Mac had a number of consultancies with us here at CIMMYT, really exploring a various range of aspects around really moving genetic resources and specific variants into breeding populations. So I'm going to give you a quick whiz through where some of that landed at CIMMYT. So the challenge, sure, everybody's gone over this ad nauseum already. I'm happy I haven't had to listen to everybody else's talk and see this 50 times. We all know the challenges. Yeah, we've got to produce more with challenges of climate change, conflict, cost of living, issues around equity, challenges around regenerative agriculture or regeneration of natural ecosystems, etc., etc., etc. Yeah, we're all big kids and know this space. Now, also, there are no magic bullets. Yeah, wicked problems require multifaceted and packaged responses. And so I'm certainly not here to tell you that genetics is everything. We're all aware that genetics is one element in among a whole smorgasbord of options. So that's precision ag, mechanization, improved agronomy, husbandry, seed systems, policies, keep adding to the list. There are many things and many levers that we need to be pulling and influencing in order to even start to address some of the challenges that we're facing. I heard John mentioning the breeder's equation, so I am slapping it up here. So yeah, all of us, I hope, around the world joining here and certainly sat around in NIAB are very, very familiar with this equation. So that we're aware of some of the elements and levers that we can pull to try to improve genetic gains for particular targets or for target product profiles themselves. And in here, we focus on genetic variants. And so, yeah, just up front and central, breeders do not lack general genetic variants for the broad traits of interest that they're working on. Yeah, there's enough genetic variation in most elite by elite crosses to mine that for a very long time and still get good genetic gain. What breeders often lack is specific variation for detailed traits of interest. That could be a particular suite of alleles for a disease resistance. It could be something that affects flavor, taste, processing, etc. And there are requests that we get continuously from the community about new variation or novel variation to their current breeding pools for abiotic stress tolerances. And really what we've been trying to focus on in the last few years is how to identify these novel genetic variants. And when I say novel, it doesn't have to be going back to a wild relative. This could be something that is simply exotic to your current breeding pool. It can be another elite that's outside of your particular TPE that you're breeding for. So how to identify those variants, yeah, it's valuable to identify them. And more importantly, how to deliver that genetic variation to breeding programs. And so the Genetic Resources Program at CIMMYT that I currently head this has been our core focus in the last few years. And here, yeah, we can talk to simply inherited traits. There are many tools and suites coming along, not only for forward breeding approaches and other kind of breeding approaches, but gene editing is a potential solution here, another tool that we can pull out of the box. But what I really want to focus on are those traits that are more complex, so oligogenic, polygenic traits. Once you get more than 5 to 10 QTLs, doing anything in a forward breeding approach gets really difficult in terms of the population sizes and just the logistics you've got to manage to try to capture that random recombination in a comprehensive pattern. When we look to the CG centers themselves, and I know, yeah, John and I had a good discussion about this last week in Rome. Many of the CG centers hold international germplasm banks, and those germplasm banks aren't owned by us. It is material that is held in trust under the International Treaty for Plant Genetic Resources for Food and Agriculture. And we do get a lot of requests into the bank. Please send me an accession that is drought tolerant, is heat tolerant, has good tolerance to salinity. Keep adding to that list. And at the minute, when you look at the banks, it's really difficult to pick anything in there in a more clinical manner that would be useful for breeding. So I use the analogy of a supermarket. So if you walk into the vaults of the banks, you've got packets on the shelves and yep, looks great, huge array of potential novel diversity that you could mine. But it's like walking into a supermarket where all of the products on the shelf are unlabeled. It's almost a random guess as to whether you get a can of tuna or a packet of dried tomatoes. Yeah, you really can't go in and make a clinical selection. And so we're trying to add information into the bank and really look at how we go in and make selections for what is potentially novel variation in a cheap and cost-effective manner. And this brings us to the allele mining initiative. So this is a project that is led by CIMMYT, but by far we're not doing all of the work here. We're working on maize, obviously, it's CIMMYT. We're working on sorghum, rice, cowpea, common bean, and cassava, with many partners in the CG and also a number of US universities. And this is funded both by the Bill and Melinda Gates Foundation and FAR in the US. So, yeah, germplasm banks are reserves of native variation for adaptation. Yeah, we all understand those bottlenecks that have occurred both during domestication and modern breeding. Now, in all of this, yeah, I'm not going to look at this through a rose-colored lenses here. Yeah, breeding has done an excellent job of removing a lot of crap. Yeah. Many land races, breeders quite respectfully wouldn't touch with a barge pole or somebody else's barge pole. Yeah, that linkage drag and the deleterious variation that comes through is massive. But in that selection, either through not sampling materials broadly enough, or because of odd linkages, or just the randomness of recombination, we've got a situation where some favorable alleles have been left behind. And really, it's a question of, yeah, is it worthwhile going back and looking for those? And if so, how do you do it strategically? So here you have some of the samples of maize diversity we have at CIMMYT. Very pretty, looks very nice. Now, thankfully, for many of the collections that we have in the international system, we have reasonable passport data for these materials. And just like your or my passport, in many cases, it details the collection site of origin of these materials. And this data can be really monetized in quite a valuable way. So using the passport data, using those geolocations, we're able to go back through varying levels of climate records. And so we can try and pull out a reasonable time window during which those collections that have geolocation data have really grown and evolved. And so you're sampling both natural variation, adaptation to the environments themselves, but also you've got farmer selection on top of that. So yeah, I don't know a farmer out there who's going to pick something just because it looks pretty. They're going to select things because it's producing grain. There is reproductive fitness compared with other materials in the sites of interest. So these collections, we've genotypically characterized them. In the case of maize and wheat at CIMMYT, we're pretty much through the banks. Other collections are adding information now. So you take this information, you've got your genomic data, you've got your environmental variables of interest. That could be precipitation, it can be temperature. All of these we try and right size to consider the growing period in the particular locations where these materials have been collected from. And you run a GWAS. It's not rocket science. Bog standard GWAS. And yes, we start to pull out variation of potential interest. The site you see here, this Manhattan plot, this is an area on chromosome 1 of maize. Sits right on top here of a potential glutathione transporter, a putative glutathione transporter, known to have a response in a lot of the physiological responses we see to abiotic stresses, heat and drought stress. And underneath, you can see a little graphic where we've taken an existing trial. We've got genotypic data from the entries in that trial. There's about 1,500 materials here, and we've classified them simply on their genotype at this one locus. And as you can see, if you are heterozygous or you're homozygous for the favorable allele, the capital A allele, we have a boost in performance in the field. And we tend to see this across the board. So you are able to use this information without initially having to phenotype anything, to pull out lists of alleles that are potentially valuable in breeding. Great. Okay, now I've got a long list. I've got thousands of potential sites. Now what do I do with them? 7 to 8% of the variation that we see here currently, as we're looking, is private to the land races. So when we get the lists of potentially favorable alleles back, the first thing that we do is go to the existing elite material. Now that is CIMMYT elite material, it's elite material from other CG centers, it's any elite material we can get our hands on. XPVP materials from the US are also included here. And so if these favorable variants or potentially favorable variants are already in the elite material, fantastic, we leave them there, not to worry about it. So we focus on those things that are really novel. And the question is, how do we start to bring those forwards into the breeding germplasm? Now, that's all good and interesting. There's another thing that we've been looking at. John talked to genomic selection, genomic prediction. And yeah, we have been looking at that, and I can talk to that in a little bit. But we've also been using this information in another way. So you can get your climate data, proxy for a phenotype. You've got your genotype information, and you scrunch it together in a training panel. Okay, nothing novel here. But instead of asking the question, here I have an untested genotype. Yeah, what do I think is its performance? We turn that around and we say we have an environment that we're interested in. Now, this may be a particular TPE that we're breeding for now, or it may be an environment 20 years in the future. Okay, what is the environment in the East African highlands going to look like for maize in 20 years' time, bringing in some of these climate models? Pushing that information through the training panel with the kind of climate data that we pulled out from that particular location or that particular TPE, you derive a putative genotype, a predicted genotype, or an idiogenotype. Now, obviously, this thing doesn't exist in nature, but we're asking the question, what genomic contributions would potentially give us higher levels of fitness in this environment? And when you start to look at this, again, using real trial data, it's pretty good so far. So here we've taken this idiogenotype that we see in the bright blue. We've measured the distance of actual things we've evaluated in the field. So measured their genetic distance from this idiogenotype. And as you can see here, as we move across that x-axis, you get materials that are less and less related, are more and more genetically distant from this idiogenotype. And on the Y, we've got a proxy for performance. In this case, it's grain weight, yield. And you can see as we get further and further away from this idiogenotype, we start to lose fitness in the field. Yeah, the adaptation of these materials to that particular environmental niche is not as good, and you start to see that in reproductive performance. So we're now using this to test this case as we move into more elite germplasm. Now, obviously, the elite germplasm is not as closely related to this training panel as a set of land racers may be. However, the set of land racers does represent in there pretty much all of the variation that we're seeing in the elite germplasm. The elite germplasm has come from this set of land racers originally. And so by having a broad representation and really capturing at a reasonable frequency most of those alleles, we can start to use this information in a different way. Right. So we can use this information to move from kind of a supermarket approach to a future market approach, identifying not only those variants of higher potential for adaptation, those novel variants that didn't make it through the breeding bottleneck, but also looking and considering how may we want to reconstitute the makeup of breeding programs to start now moving in a direction that we anticipate we're going to need to be in 10, 20, 30 years' time. So taking that aside, and here is a slide I have willfully pillaged from Keith Gardner, thinking about the actual introgression of this variation into breeding. Typically what happened in the CG is this. You'd have an unimproved material. Okay, here is your exotic. It carries what we think are favorable alleles for something. Go ahead. And the breeders would take that landrace and throw it straight into their existing breeding pool. Okay, so John has talked about the decoupling of breeding. This is what we're doing now, tending to look at recycling separately to actually looking at product identification. But this whole kind of willful, let's just chuck a landrace in there and see what happens, yeah, fills us with utter fear nowadays. Yeah, I mean, as any breeder would say, understand, yeah, it's going to take your breeding population back by decades. So there is a focus now on really providing semi-elite material as bridging germplasm, as trait donors, yeah, pick your favorite term for this, but something that is semi-tamed. Okay, so we're not throwing the tiger into the mix. Again, you haven't got this beautiful street cat coming out, or this beautiful Siamese on the end. It's something a bit shabby and in between, but it's not necessarily going to try and take your arm off when you're breeding with it. There are many different schemas that have come up around this space. So yeah, this was one allié that talks to this pre-breeding and this bridging phase. There's many schemes that have been published recently that many breeding programs have been using quite effectively for over a decade. We looked at how to best initiate these populations. Yeah, we had a publication here with this unusual suspect. I don't know who this dodgy bloke is here, but really looking at how we best try and constitute and select these materials. But we hadn't always taken that forward into practical, okay, how do you really do this in an effective way over and above the standard phenotypic approaches that we have been using reasonably successfully? And really, I frame this as getting the wheat from the chaff. And so this is the culmination of some work that we did with Mac, really looking at how to speed up that process. We know we can do this through phenotypic selection. Yeah, we have been doing it. This is where a lot of the very good trait donors have been coming from the CG system. And this has been happening since the 90s, yeah, 80s, 90s. It's not new. but really looking at how we start to layer in genomic information and ask the question, can that help us really accelerate this process and become much more targeted? And this was a publication that a student with working with Wayne and Ian, and then pulling in data and paradigms that we were pushing forward, published a few years ago. And this is just one snippet coming out from it. So yeah, go back and read the publication to your heart's content. But essentially, we pulled out a suggested approach. So origin-specific genomic selection, whereby instead of just throwing a normal genomic selection process in here, where we know what are you going to pull back out, you're going to pull out the elite backgrounds in any kinds of crosses, whether the biparental crosses or whether you're doing back crossing here. That elite parent has simply a massively higher genetic merit than your exotic parent, and anything favorable coming from that exotic is just going to get swamped out. And so, yeah, not rocket science here conceptually, but thinking about how you apply weighting within this system to be able to appropriately reconstitute the value of the elite parent. Yeah, breeders don't want bridging germplasm that sucks, but equally to allow those favorable alleles from your exotic parent to come forward. And so what we have here, this is just one of the graphics here. This is a BC1 population. You can see the QTL distribution in terms of genetic space. And what you've got here is the breeding value over a number of generations. And you've got the proportion of favorable QTLs that are coming forward from the primary parent. So here is your elite parent versus your exotic parent. And as you can see with the origin-specific genomic selection versus standard genomic selection, you're pulling different contributions forwards. So you're tending to select more alleles from the exotic parent than you are from the recurrent parent, that elite parent, which is, yeah, what you'd hope you'd do if you do origin-specific genomic selection. But here we're really not taking a big hit in breeding value. And this is one of the key important things for that bridging germplasm. It has to be rigorous and robust in terms of its overall breeding value. So looking to develop tomorrow's varieties today, institutionally, genetic diversity and germplasm bank collections, when we explore them clinically with modern tools, good partnerships, they are a valuable resource in the efforts to mitigate some of the impacts, in particular of climate change. We know they're an ultimate supermarket of ingredients. Yeah, if we really want to go back and find genetic variation, my son always asks, what would happen if there was a zombie apocalypse tomorrow? Well, I'd be in the bank, because A, the zombies can't get in there, and B, we've got variation to start again. We can look at the development and delivery of seed-based genetic insurance to farmers and use some of this genomic foresighting to help guide where breeding programs potentially need to be moving in climate change. And I hope I've demonstrated that there are approaches that exist to guide these pre-breeding steps. Now, I'm not saying that absolutely every program in the world needs to jump into this. It would not be a good use of resources. There's a lot of standing variation that is there within the elite pools to mine, but where they become limited and we need to strategically go back and look for variation, we've got some tools now to help us do that more effectively. So yeah, reflecting back on Mac's contribution here, this is something that he brought me as a birthday present when he came out to visit once, which is still stuck up on my wall. So looking at how to leverage evolution, and here a great example of how we've probably evolved scientifically, or maybe even haven't evolved scientifically when I look at some of the daft stuff that we still continue to do, is really moving forward and really leveraging evolution to our advantage. So I don't think any of us are quite the Einsteins here, but we're, yeah, hopefully I'm no longer still dragging my knuckles along the ground when it looks to mining this variation appropriately. And so, yeah, thank you. That's it from me.
Thank you so much, Sarah, for that presentation and your reflections and Mark, which I think are so appropriate and well received. So a couple of minutes for questions from the room. Want to go question?
Sarah, did you say something about where in the world most of the useful germplasm in the gene bank is coming from? So is it coming from particular ecotypes or is it randomly distributed?
Okay, no, great question. And no, I didn't go into that, John. So when we look at the variation itself, you do have some spaces which are pockets. I mean, if I reflect on the maize bank here, most of the collections in the bank come from the primary and secondary centers of diversity. I mean, yeah, that's what banks should be representing. But you tend not across the board. If I look at 100 to 1,000 variants of potential favorable effect, you may get 10, 20% of those that are coming from relatively discrete pockets. But overall, you don't see massive bottlenecking. Yeah, I mean, you do see some alleles that may be coming more from that Peruvian complex where you did get a genetic bottleneck post initial distribution of maize. And so that's more representing new evolution that hasn't made it back and repatriated to Mexico in the flow of the crop itself. But you tend to see more broader representation of these particular variants. So not always from discrete pools. Yeah, it's more broadly distributed than initially I thought it would be.
Are there any questions online? No. Any more questions from here? Sarah? I have one, Sarah, and that is, I guess, how much of the variation from a gene bank could you capture in a magic-like population?
It would depend on what founders you'd used and how many. It's we can capture, if I look at the 25,000 land races that we have in the bank, I can capture 96% of the variation mass or minus in 1,000 accessions. Yeah, pretty well. Now, you're not going to be making a magic population from 1,000 founders. We haven't scaled that back down to smaller numbers. And the returns drop off pretty quickly when you're looking at things like maize, which is I mean, as we all know, it's an open pollinated species itself. There's more variation between, if you partition the variants, there's more variation within an accession than between accessions. And so it's, yeah, thinking about using individual plants in that crossing, it gets quite complicated. In a species like wheat, you should be able to sample fewer things and still capture reasonable amounts of the variation. But we've not looked at structuring that down into that level. I think we need rather a lot of magic populations to try and cover that broad representation of variants that we see in the natural diversity.
Thank you very much, Sarah. Two questions. Phil, and then behind you here.
So just building on that, would it be possible thematically to intercross some of the lines that you know are carrying high value rare alleles? So instead of just using one line as a source of a particular allele, you're pyramiding them. So you've got maybe one line that's carrying 7 or 8 different sources, and then that becomes a founder. And then so if you're there's a pre-breeding step before you start your magic into crossing. So you're starting to pyramid these rare alleles together in a single line. That then becomes the founder for a multi-parent population.
Yep, and we'd looked at doing that. So some of the things that we did with John and Gregor and the simulations that we ran quite a few years ago now, looked at different strategies, kind of running up to pre-breeding proper. And a lot of those were pyramiding strategies. So when, and just to give another reflection on that, at the moment when we are selecting materials through allele mining that we're using actively in this pre-breeding approach, we initially get, as I say, the list of alleles. We focus down on those that are private to the land races. And then I go back into the entire germplasm bank and really look just at those particular alleles of interest and say which land races concentrate the highest frequency of these potentially favorable alleles. Yeah, and that's looking across the whole allelic profile that we have. And yeah, you get some, okay, this material from the Andean region in Peru, this is super good, but I know there's not a cat in hell's chance that we can cross with that easily here in Mexico. Yeah, you just don't get nick, you don't get a breeding cross. And so we go back and use our understanding of adaptation and what will grow effectively here, pyramiding in, yeah, how do we capture the most in the smallest number of accessions to start with before we do those breeding crosses. At the moment, we're not doing the intermating beforehand because I want to see if something is actually bringing value forward quickly. Yeah, I can do an awful lot of intermating and then start phenotyping things and think, oh, no, actually this sucks. And so we do that crossing early on and that testing early on with much more of a product development mindset. And then as we start to clean these backgrounds up and really get this variation into something that behaves better in the field, then we can really start evaluating that. But there's many ways you can skin a cat. And really the question is whether you're more focused on knowledge generation or we're more focused on product generation. And certainly product generation is the key focus here, with knowledge generation as a secondary, but still very important feature.
Thank you. Hi, Sarah. My name is Karis. I'm a wheat pre-breeder. I think like if we look at it, the history of breeding, there's been a couple instances of pleasant surprises where we don't see the phenotype in the accession. But once it's incorporated into the elite germplasm, you see a phenotype. So it's back to like what John was talking about, these anomalies. Do you think in the future there's still room for these kind of like speculative explorations of diversity, provided there's not a negative impact? Like it doesn't sound strategic to be speculative, but could you share like your feelings about this?
Yeah, no, and it's a great question. And it's a question that we banter around a lot. So I have colleagues who've worked their entire lives in that speculative space, and yeah, serendipity happens. You get some randomness with a breakage of a particular linkage block or something else, and yeah, you find something you didn't expect to find. When you look at the bulk of breeding, yeah, that is not the case. We always remember the exceptions because they're exceptions. They're absolutely and utterly not the rule. That isn't to say that there isn't a space to do something speculative, but you've got to really look at the return on investment. Now, when we're considering the bulk of genetic variation, yeah, we're able to make gain for most things. And you're able to make pretty good gain, and often more gain than we think we're able to make if we use the appropriate levers and selection approaches. And so I think that has to be the biggest workhorse by far. Then you've got that space which is a little more speculative. You're looking for new variation, but you're doing that in a very structured and strategic manner. And we tend to be moving to be much more ruthless in terms of what we follow through on. So we've got very clear stage and gate processes. We've got very clear KPIs that really tie into the breeding demand. Something doesn't look like it's performing, it's thrown in the bin. We're not hanging on to it. But there is some space in that speculative area. Now, there are other tools and resources that we can use to bring in to really look at representation of diversity that we're bringing in, novel pockets of diversity that we've never sampled, etc. So it's not just to say, now in the speculative approach, go and grab something at random and run with it. There's still ways of trying to stack that pile of cards so we're more likely to build a tower. But at the end of the day, it is in any breeding program, research program, about looking at those priorities investment and really getting a reasonable portfolio. So in terms of the time distribution or resource allocation, that speculative stuff either needs to be done in completely different institutions. Yeah, let somebody else work on the speculation. Or if you're doing it in your own program, it needs to be with very ring-bound, very defined, and very framed investment that is not eating into the bulk and the core of your main breeding program investment. So it's the amount of money you're willing to lose and throw away, because the likelihood of success is low, but the impact of that success may be massive, yeah, if you can find it. But it's not something that's going to be your main food source, if you want to think of that from investment for years to come.
Thank you very much, Sarah, and thank you very much for your questions. I'm all in favour of pleasant surprises. I think look forward to having more pleasant surprises. Thank you, Sarah, for joining us. And we'll bring the session this morning to a close. Thank all the speakers. I think we're about bang on time, and we reconvene at 1:35.
Correct.
Thank you very much, everyone.
Yeah.